now that I'm recording, I can start on a diatribe. The cavefish genome reveals candidate genes for eye loss. You're not necessarily geneticists or genomicists, but if you follow genomics Twitter, which you probably don't, which is why I'm saying this up front, people are starting to mock genome papers because the title of almost every genome, we assembled the genome of a species, it reveals something. So every genome paper basically, it has the word reveals in it, because that's the best you can do. Like, well, okay, we got the sequence of the DNA, what's going to be the point we're going to tell about this paper? Well, we're going to look for signatures of something or evidence of something. So now people starting to mock papers that say that their genome is going to reveal something because, on one hand, it's never an experiment, so it never proves anything if you just have a genome sequence. On the other hand, every genome sequence reveals something, so it's kind of a ridiculous title. Anyway, but this was back when sequencing a genome actually meant something, 2014, especially a fish genome sequence, because fish genome sequences tend to be a little bit larger on average than some other species. But, so this is a group of faculty members, Cliff Tabin and Jean-Nicolas Wolff and, oh, I thought there were more on here. There were a number of faculty members on this paper, all of whom study fish, and many of them study cavefish. And the reason people study cavefish is because of eye loss. So they're going to use this genome sequencing paper to look for evidence of trying to figure out, ask the question, why is it that these fish, how is it, not why, how is it genetically that these fish lost their eyesight? Before I get into the biology of the cavefish briefly, if you have any comparison, we've seen this a lot, this is evolutionary developmental biology. We're looking for genetic differences that explain a change in development, like development of eyes or not. So at this point, we should be able to come up with a pretty comprehensive list. We're about halfway through this semester? Almost. Wow. We should be able to come up with a list of, if you're going to look at genes or genome sequences, what are the sorts of things you would hope to find that would explain the difference, like the presence or absence of eyes? What sorts of, in the DNA sequence itself, or related to the genes, what sort of experiments have we seen so far? Okay, so knockout, so that's an experiment that would look for absence of a gene causing absence of a trait, or presence, change in a trait. Okay, so you could look for gene presence absence. That is, if you had the sequences of the cavefish on the top that has eyes and the cavefish on the bottom that doesn't develop eyes, and you're going to compare those genome sequences, you could look for which one has a gene that the other one doesn't have, or vice versa, and imagine that that difference might have something to do with this difference. Might. You'd have to do experimental tests to prove whether or not a gene had that sort of a role. What else? So the gene presence or absence in the genome, that is gene gain or loss. You could also add copy number. We've looked at genes that duplicate and maybe duplication. So we could also look at gene copy number change from one genome to another. What if there is a gene that's important for this difference, but that gene is present in both genomes? Okay, so gene expression, which we've seen a number of different types of gene expression. We've seen level, that is a gene expressed or not? Gene could be there but not turned on. Maybe a transcription factor is missing or something. So it could be gene expression on or off, or amount, how much of the gene is being transcribed or not. Also, related to gene expression, not just amount, but okay, so gene, but that will change physically the amount of gene expression or other aspects of gene expression. So you're on the right track.
The timer.
Time, so temporal. So it's not just how much of the gene is turned on, when is it turned on, and where in the body it's turned on. So where is this gene transcribed, and then in time and place, does that change between the two species? Any of those sorts of changes might hint at transcription factors or changes in transcription being important in this difference. So even if all the genes are the same in the two species, there could be subtle differences, and those would be things like, for the example of a transcription factor, although it doesn't have to be in a transcription factor, mutations or DNA sequence differences. So it might be that there are transcription factors that are important for eye development in both of these species, but one is a wild type and one has a mutation. So the genes are still there, but there's a sequence difference in the gene that's important that causes that effect. So those are some of the things that we're going to see them talking about on Wednesday. Now we've got a genome sequence, and they're also going to do transcriptomics, which means looking at RNA sequences. So they're going to try to have the catalog of all of the DNA from a blind cavefish, and they're going to try to have the transcriptome, that is the set of all of the RNA molecules that are expressed, transcribed from the genome, and look for these sorts of correlations. I would like you, please, to pay attention to whether or not there's any causation. Are they doing any experiments that are experiments that are experimental? Are they going to do any experiments that prove the role of any of the genes that they find that they think there are that may be incorporated or involved in eye loss? Something to think about for Wednesday. There's a lot of slides in here and not a lot of time, and I'm not one to rush, as you know at this point. So we won't talk about all of this. I will focus mainly on cavefish, the concept of QTL mapping, although that's not actually part of the paper, but it's data on which the premise of the paper is based. Previous studies that were quantitative trait locus mapping studies. So I'm going to go briefly into that. They're going to use the term linkage group, or LG, a lot in this paper, so it may behoove you to understand what a linkage group is, and we'll talk briefly about pleiotropy. Transposable elements, there is some small aspect of this paper that has to do with transposable elements. On Wednesday, let's talk about if that was actually a relevant part of the manuscript to the story of the manuscript. And we've already seen, they're going to use these techniques. We've already talked about quantitative PCR in this class. We've a little bit talked about RNA sequencing, but that's a simple concept. You take RNA out of a cell and you sequence it. And we've seen in situ hybridization before. If we have time at the end, I'll demo and I'll ask you to follow along with your devices in an exercise doing BLAST. Some of you have done this before in other classes, some of you haven't, but it's good. It's a technique they just mentioned briefly, but it's a good technique to know about for evolutionary developmental biology. So that's where we're heading today. Talk about the biology of cavefish, some genetics experiments, and then we'll end with BLAST. The Evo-devoists love the situation where the same trait has evolved multiple times, like hermaphroditism in Caenorhabditis, because we can answer those questions like, do the same genes cause the same traits all the time, or are there different ways, different types of mutations to get to the same endpoint, the same phenotype or the same trait? And so, like my favorite worms, loads of biologists around the world love studying cave dwellers because even though these are very distantly related species, I think you can appreciate, some of them have limbs and some not, for example, they all tend to have the same sort of traits evolve. When animals live in caves over a long period of time, what happens? Tell me.
Loss of pigment.
They lose their eyes and they lose pigment. They stop developing eyes and they stop producing pigment. Why?
Conserve energy.
They don't need them.
Right. They don't need them anymore. What does the pigment do for a non-cave dwelling organism?
Helps UV.
It could be UV protection, if it's melanin, for example. Helps block ultraviolet light from hitting our DNA.
Predation, mating.
Yeah, camouflage, mating, they're signals. It's communication. If you don't have eyes, you can't visually communicate, so what's the point of producing pigment? It can't be selected for or against. Predation isn't happening on you because you've got one particular type of pattern or another of pigment if nobody else around you can see. right? No predators, no mates. Well, what's the point of having pigment? So this happens repeatedly in lots of different types of species. And the same is true in the genus Astyanax, which is a large genus of fish that live in a bunch of different subterranean caves in central Mexico. And most of these, like the evolution of hermaphroditism multiple independent times in Caenorhabditis, lots of these cavefish populations have independently invaded separate caves and all then evolved lack of pigmentation and loss of eye development. There's this great natural, repeated, replicated experiment. So you can ask, what happened in all these individual caves to these different populations of the same species or close relatives, closely related species of fish? How did they all become pigmentless and lose their eyesight? And based on fossil records and other things that I'm not familiar with, but maybe the two of you are more familiar with, and so I'm glad you're presenting this paper, they know at what points in geological time cavefish invaded these caves. And it has, of course, to do with sea levels and whether or not fish could get into these caves from other sources. So for example, these caves are near rivers. If the river levels and the ocean levels are high enough, then fish could swim between caves, but at some point ocean levels dropped, isolating the different fish in these different caves. So this is a really important thing, which I didn't put in here, but is really important when you study repeated evolution like this. You need to know, are organisms moving in between caves? Because if individuals are able to migrate between the different caves, then you might expect that they're all sharing the same mutation that causes them all to lose eyes. If they're all breeding with each other, then they're more likely to share one mutation that caused them all to have the same sort of eye loss and pigment loss. But they know from geological history and geology that these caves are isolated. So fish got trapped in these caves and then they weren't able to get out. There's water in the caves, but there's no water leading into or out of the cave. So they're independent populations. They've been there for millions of years, evolving independently. And so who knows how long in terms of numbers of generations it takes to lose eyes. But we know at present day that they have, because their close relatives are surface cave fish, Astyanax, that don't live in the caves, that live in the rivers. They have eyes.
Quick question. So does that mean they're like saltwater fish in a cave? Or is that
These are all fresh. As far as I know, they're all freshwater. That they were freshwater fish living in rivers near the caves, but at some point, some fish were introduced into the caves, which were also freshwater. I probably shouldn't have said ocean.
I just thought sea level.
That's what I was thinking. I shouldn't have said sea level or ocean. I was thinking more sticklebacks, which is the case of sticklebacks, which was when sea levels were high, they invaded lots of lakes that were up in the hills. And then when sea levels dropped again, that stranded marine sticklebacks, and not to get too off topic, marine sticklebacks into freshwater lakes, and those marine sticklebacks evolved to tolerate fresh water. But that's not what happened in cavefish. But that's a different story that's also Evo-devo, and maybe relevant to who's doing the stickleback paper. So we'll talk about that then. The gain and loss of armored plating in stickleback fish. Right, so development of cavefish. This is a surface population on the top of each of these panels and a subterranean or cave population in the lower part of each of these panels. And eye development never really starts. They do say they make an internal eye that's covered by skin. So they do sort of develop kind of a rudimentary eye, but it never fully develops. And so you don't see an eye. It looks just like flesh and skin. But there is a rudimentary eye in there. It just doesn't fully develop. So this is the cavefish and a close relative from the surface population. And this has happened so recently in time, order of millions of years is a long time, but not too bad, that the scientists, and this is not from this paper, this is from other papers on cavefish, know that you can cross surface and cave populations and get hybrids. And so one of the first types of experiments that scientists did that were studying the cavefish was, yeah, what happens when you take an eyeless fish and made it with an eyed fish? Do you get eyeless fish that are offspring, or do you get fish that have eyes that are offspring? So in each of these, let's just focus, although it's the same pattern in both of these, so we could look at one or the other, presence or absence of eyes and amount of pigmentation. Each row and each column is an abbreviation for a different population of cavefish. And so you can see on the diagonal, Pachone by Pachone, that's a cross of two members of the same species. The diagonal is crossing one population with itself, looking at, so to speak, purebred populations. And you see that most of these, when you cross the cave populations with each other, they develop small internal eyes. So they can't see. And there are a couple that develop intermediate sized internal eyes. So within each of these populations, they don't develop eyes, but what happens when you cross different populations together? Anything off diagonal? Are there any cases in which you can cross two cavefish populations, neither of which have eyes, get offspring that have eyes? Sorry, yeah, it's hard to see because it's small, and you can have this if you want to, it's uploaded to Canvas, but large external eyes is that color. So that's the same cross. Pachone by Rio, I don't know what the S stands for. There's one cross you can take of two blind cavefish, and you mate them together and they make normal fish that have eyes. Well, I don't know if they're normal fish. They have eyes. They can use them to see. So why does that tell us that loss of eyes is genetic? And the same is true for pigmentation, by the way. Extensive pigmentation here in black. You can set up multiple different types of crosses between different combinations of populations and get pigmented fish when all of the parents are albino, unpigmented.
That means they still have a sequence, it's just being turned on.
Right, it means that the DNA has the capability of doing this, of making eyes and making extensive pigment.
It's just not being expressed.
It's just not being expressed, which suggests, the upshot of this is, it suggests that different mutations in different populations cause eye loss and pigment loss. So if the Pachone population has come up with one particular set of mutations that prevent eye development, then by themselves, if you made two individuals from that population, they wouldn't have offspring that have eyes. But if the RS population has a different set of mutations in different genes that make them not have eyes, if you make a hybrid between these two, the Pachone fish donate the wild type copies of the genes that those fish are delinquent for, missing, and vice versa. The RS fish have probably wild type copies of the genes that are mutant in Pachone that cause them to produce eyeless fish. It's a genetic approach called complementation mapping, if you want to know the details. But it suggests that this loss of eyes is not environmental, that it's genetic, and that it involves multiple sets of different genes in different populations. And the same for pigmentation. And relevant to pleiotropy, which is a term that's hard to define. It's simple to define, it's hard to appreciate, I think. What we're going to be talking about pleiotropy a lot on Wednesday, I think, is note which fish have the large external eyes, where they are located on that diagram. And note which population also has extensive pigmentation on the right side, the black squares. Is there any overlap between the two?
Yes.
Right, the lower left and the upper right corners have combinations where both of the parental populations are eyeless and pigmentless, but crosses between them produce not only eyed offspring, but pigmented eyed offspring, which could be evidence that there's a single gene that you can mutate that causes loss of eyesight plus loss of pigmentation at the same time. That's the concept of pleiotropy. That you can have one gene that has multiple effects. What single gene, you can make one change, like a deletion or a point mutation, that will have numerous different types of outcomes in the organism. So one of the things that these authors are interested in is that question, is there a relationship, a genetic relationship between pigmentation and eye development? so that if you lose one, it's likely, or at least possible, that you lose both at the same time. Because wouldn't that be really cool? Because then you get rid of two traits that you don't need with a single mutation, maybe, which would be pretty advantageous. So in terms of figuring out what sorts of genes control traits, genetic mapping is a common approach. It's a technique that's been used for almost a century. I'd wager, maybe not quite that long. So here's the idea, especially as it involves pigmentation. Say you have two different individuals, and I know you can't see the letters, it's totally fine. You don't have to be able to zoom in, although I could zoom in. Let me help. Technology's great. So we have two different individuals, and so we're going to assume that there's a single gene that is gene A. And it's got the lowercase version, which causes light pigmentation. So each of these A's contributes, this is a diploid individual. So they have two copies of gene lowercase a. Each of those that version of the gene doesn't make any pigment. So if you're A over A, if both of your copies of the chromosome have this version of the gene, the lowercase, then you are lightly pigmented, or you don't produce pigment. And you have capital A, two different versions of a gene, each of which produce pigment, so you get a very dark individual. If you cross that individual with a very light individual, then at fertilization, the offspring gets one capital A from this parent, say in the egg, and a lowercase a guaranteed, because there's only lowercase a's over here, in the sperm. So little a and big A meet and create a heterozygous individual. They have a little pigment producing gene and a lot of producing pigment gene. And so they make an intermediate amount of pigment compared to the parent that had two copies of the capital A, which made, presumably, in this cartoon, twice as much pigment. So you've got 2x pigment, 1x amount of pigment, and 0x pigment. And then if you took two siblings and mated them together, what are the possible outcomes of that cross in terms of amount of pigmentation? You can have capital A, you could have lowercase a, or you could have more capital A, lowercase a, heterozygotes, which is that diagram. So you start with diversity, you get an F1 generation they're all identical in pigmentation, and then in the second generation, the F2 generation, you get diversity. You get little pigment, no pigment, a lot of pigment in different individuals. This is what happens with just two genes controlling this trait. Two alleles rather, of one gene. You can either make a lot, make a little, or if you're hybrid, if you're heterozygous, you make an intermediate amount of pigment, in theory. So that's with one gene controlling pigmentation. So this is Mendelian mapping. Mendel studied a bunch of different traits in peas that had one gene that controlled a trait. So you'd have a gene that controls pea color, green peas, yellow peas. There was a different gene that controlled pea shape, wrinkly or round. Everything was binary. One gene has two different alleles, two different versions. One that makes round peas or wrinkly peas, yellow peas or green peas. I don't remember what the other traits were. I should, since I teach genetics. So that is if one gene controls the trait of interest. That is usually not the case. And this is what my daughters are asking me all the time. Is it really true that blue eyes is recessive? No, it's probably caused by a combination of genes. Is earlobe attachment a Mendelian trait? Is it controlled by one gene? No, it's probably caused by multiple genes. Most traits are caused by multiple genes. There are very few traits where a single gene is controlling the entire trait. So enter quantitative locus or QTL mapping. This is a way for scientists to try to figure out, the point of this is to try to figure out what regions of genomes contain genes that all in combination contribute to the production of a trait, like pigmentation. Pigmentation is not usually controlled by a single gene. eye development is not controlled by a single gene. So in this simplified version, now we're looking at a combination of 3 genes, gene A, gene B, gene C, all of which independently produce some amount of pigmentation. So now that instead of looking at 1 gene, we're looking at 3 genes, you can still create hybrids that have an intermediate amount of pigment, just like the first story. How does this story differ? Now you've got 3 different genes, each of which have 2 versions, capital and lowercase. What happens when you mate these F1s together? There are a bunch of different combinations of which versions of the 3 genes come together. Right, there's 64 different combinations, not all of which are different. So those, for example, all contain 1, 2, 3, 4, 1, 2, 3, 4. Each of these in this box contains genes, or combination of genes, if they're additive, that produce a 4 out of 6, if you will, mathematically description, 4 out of 6 pigment. These are 2/3 of the total amount of pigment. If you get 5 out of 6, those are those combinations or those combinations, and then the ultimately pigmented combination would be having all 6 capital letters that make as much pigment as possible. So now we're seeing shades. It's not pigmented, not pigmented. It's not pigment, partly pigment, not pigment, like with 1 gene. With 3 genes, now you get this array of different traits. And importantly for quantitative trait mapping, what we do is, the first step is to create hybrids. So you can imagine crossing a bunch of different fish and getting a bunch of hybrids that have intermediate levels of pigmentation. The goal, again, is to figure out what genes or what regions of chromosomes contribute to pigmentation. That is, where are these 3 genes located on chromosomes? So one step, then, you have to take after you make these hybrids is to phenotype them. You measure how much pigment they have. That is the trait we're trying to study. Where are the genes that control pigmentation? A quantitative trait, then, is defined as one that is controlled by multiple genes. And we know that when we see this diversity in hybrids. So if it's a Mendelian trait and there's just 1 gene controlling the trait, you'll see 3 different versions of the trait, which we saw on the previous slide. It would be no pigment, intermediate pigment, or pigmented. When you start seeing shades, or a continuous distribution of different types of, in this case, pigmentation, right? There's one that's very little pigmented, there are more that have a little bit more pigment, so you start to develop a bell curve. When you see this in traits, in hybrids, that suggests that multiple genes contribute to the trait. So those are the hallmarks of quantitative trait loci, when multiple genes control a trait. You see lots of a gradient of trait, not just discrete versions of a trait, like all or nothing. So when scientists, for example, crossed cavefish and saw a diversity in pigmentation, that suggested that they needed to use quantitative trait locus mapping approach. So in fish, then, what was done in previous research by one of the authors on this paper, Bill Jeffrey's lab, they made a huge number of hybrids between surface and cavefish, and then they measured a bunch of different quantitative traits. One of them was they dissected, this is fun, they dissected a bunch of fish eyeballs. After the hybrid fish all grew up to adulthood, they euthanized the fish, they took out their eyes, and then they sectioned them. They cut sections of the tissue, and they measured how thick the different cell layers are in the developing retina. And they saw, these are then representatives of siblings. These are F2s, I think, so grandchildren, they'd be first cousins. And relative differences in the amount of tissue that develops in the eye. And the fact that they saw such variation from individual to individual in the thickness, the amount of cell tissue in these different layers of the eye, suggested that this was a quantitative trait, that multiple genes contribute to the thickness of the inner plexiform layer. You don't have to know what an inner plexiform layer is. Or the ganglion cell layer, or the retinal pigmented epithelium. So that's this is one of the most recent publications before this paper, was they did a quantitative trait focus scan. This is another example from that paper showing a highlight of a quantitative trait, which is that, for example, ganglion cell layer, the thickness is based on the height of the bar, right? It varies from individual to individual. And all of these different cell layer thicknesses in the different individuals, each one of these lines is an individual fish, each column that is. So they found that in this case, eye development, the thickness of these different tissues was continuously varying. It wasn't discrete. It wasn't like you could have 1 cell or 3 cells or 1 cell and 4 cells. There's a lot, you could have a huge range in the size of that cell. Suggests that each of these are controlled by multiple genes, just a single gene. So that's what is that? The second step, you make the hybrids, you phenotype the hybrids, you measure something, in this case, eye tissue thickness. Then you get the genotypes of all of those individual fish. That is, you basically sequence their genomes. So you know how each of those individual fish is genetically different from every other fish. And then the part where the computer comes in, which is nice because you can't do this by hand. You ask, for example, in the fish that have the thinnest, in all of the fish, let's say you studied 500 fish and 30 of them had the smallest ganglion cell layers. They all had very thin ganglion cell layers. You would want to know, if we looked just at the genome sequences of those 30 fish, is there some mutation that they share in common? Because if they have a trait in common, and if there's a genotype that's common, that suggests that genotype, those mutations might create that trait. The problem is the genomes are millions of base pairs large and there are lots of mutations, which is why a computer has to do this. So you ask the computer, can you find computer running for day and night, 24 hours a day for a couple weeks? Can you find combinations of, let's say this gene here on chromosome 1 and that gene there on chromosome 12 and that gene over there on chromosome 27. Does that particular combination correlate at all with any of the phenotype measurements that we made? Like small inner ganglion cell layer or large ganglion cell layer, or so there are an infinite almost number of combinations of traits, and then an almost infinite number of combinations of which parts of which chromosomes you look at to try to predict where genes might be located that correlate, cause, presumably, these traits. This is an oversimplified version of that sort of an approach. This is not meant to be a QTL mapping approach. This is more of a classic genetic mapping approach, but I just show it to you as a bit more concrete evidence. So say you have pigmentless and pigmented, or hairless leaves and hairy leaves, and you cross those two plants together and you get an intermediately hairy-leaved individual. And you self that individual, you mate it with another F1, or you self it. And you keep doing that until the 8th generation. This is not what they did for Cavefish, but you can do this. So you get individuals, for this one particular chromosome we're looking at, every round of mating, you sort of scramble the genetic contributions from the two different parents in the offspring. So here we have a cartoon, not a real experiment, where we can see the genotypes, red or blue, which parent did each gene come from. By the way, if red is hairless and blue is hairy, then when we see the hairy-leaved offspring, which color of chromosome should they have? That's how this works. You figure out what trait you have in the parent, and so what we're looking for then, what the computer would be looking for, is, and of course there would be more than one chromosome in a real case, but this cartoon is nice. For the individuals that have hairy leaves, where do they have blue on this chromosome? Is there one spot where all of these hairy-leaved F8, great-great-great-great-great-grandchildren, have blue?
On the lower part towards the middle, yeah.
Well, that one's got blue there. Do any other hairy-leaved individuals have blue at that same spot? Say that's the location of a gene. That individual does. Any others?
Next one.
Yeah.
Yeah, it could be that one. But now this one's got blue but doesn't have hairy leaves. And that one doesn't have blue there but does have hairy leaves, but this one's good. This is actually something that will happen in genetic mapping, by the way. We'll get there in just a second. I'll explain what's going on. But what about that individual? Somebody already said this. Who said it? What? Well, it's obvious. There's only one spot on this chromosome that's blue. So there might be a gene there that controls hairy leaf development as well. And that's also present in that individual and that individual. But not that individual that's got a hairy leaf, but is also maybe there in this individual. So the question marks are, why does that bare-leafed individual have blue there? And what was the other problem? Where was the other problem? Blue.
Yeah,
blue.
that didn't have that upper.
Right. So this is the sort of case which does occur where two different genes control hair development. You have to have one or the other, or both. This is just one possible outcome of genetic mapping. You find a situation where there are two genes that independently control a trait. This is not what we're going to see on Wednesday. But you can either be blue near the centromere and be hairy, or you can be blue near the telomere, the bottom part, and be hairy. Or you can do both, but it has to be one or the other.
That fourth one has both and it's not hairy.
Right. So that's weird.
It does super.
Yep. Maybe that means there's another gene that controls the trait. And this is what happens during this sort of process.
Or masks the trait.
Or masks the trait. Yeah. Genetics is awesome. So this paper was suggesting that this is one of the quantitative trait loci, where now we're just looking at the genotype underneath that region of the chromosome and how it correlates with leaf development. And with an addition of a second QTL. So it turned out actually that it wasn't blue that causes hairy leaf development. You have to be red at both of those loci to be not a hairy leafed individual. In other words, for those of you that are aficionados or want to be, blue is dominant. So if you have one or both that are blue, that creates the hairy leafed trait. If you have neither, then the recessive, or sort of the default, if you will, is not to produce hairy leaf. So this is not QTL mapping. This is because this is just looking at, this is looking at two loci. So it's almost quantitative trait locus mapping. But if you look at 3+, that's multiple locus mapping, which is basically quantitative trait mapping. Woo! So this is what the outcome of a QTL mapping experiment actually looks like from, again, this previous paper, the same paper I've been citing over and over again. Cavefish paper, looking at, in this case, quantitative trait loci for 4 different cell layer thicknesses. So at the top, this is where the action is. It's a QTL plot, and on the x-axis we have chromosomes. So in this case, or linkage groups. We'll talk about that next. These are linkage groups. They're basically the equivalent of chromosomes for our intents and purposes. And the peak, if a peak passes a particular threshold, which in this case is 3, the dashed line, then that region of that chromosome is statistically significantly correlated with whatever trait it is you're measuring with. It suggests that there's a region of genes somewhere on that chromosome underneath that peak. That section of chromosome looks like it has a gene or genes that control that trait. This is the outcome from what the computer does when it's thinking about looking at genotypes and traits. In other words, there's a region of chromosome here that looks like it's that genotype is significantly correlated with the thickness of the INL, whichever layer that is. And if you zoom into that part of that chromosome, these authors then are showing what genes are in that region, and then they do a candidate gene approach, which is to ask, now that we've identified that this part of this chromosome seems to be regulating something about I something, what are the genes in there that we know anything about? And do any of those genes in that region, do we know or suspect would have something to do with eye development? Like CRX, the cone rod homeobox gene. Cones, rods, those are eye cells. Homeobox, it's a transcription factor, maybe an eye development gene. So that's how this process works. QTL mapping gives you a list of candidate genes that then you sift through, not computationally, but sort of literature search-wise. What do we know about those genes? What do they do? And then to figure out if any of those genes are involved, you have to do things like knock them out. Or in situ hybridizations to see if the expression level changes from tissue to tissue, between surface and cavefish, and so forth. So for this, I will highlight a few, in particular because in this paper we're going to read, or you have already read, they talk a lot about linkage group 2. In part because it's got the tallest peak, the most statistically significantly correlated peak with eye traits. And not only does that region have CRX, which is a cone rod homeobox gene, it also has TBX2A and CryABB. That's not crybaby, that's CryABB. And right next to it is Notch, which is a signaling protein. And so we're going to hear about some of those proteins again on Wednesday. Not all of them, but some of them. So this was the paper that was the jumping off point for of Wednesday's paper. That this was already known, and then this is supplemental material on Wednesday, so I'm not going to focus on it, let you show it if you want to on Wednesday. But, oh, one important point. That previous slide I just showed you had 25 linkage groups on it, which essentially means 25 chromosomes. We're going to talk about the difference right now. This study from Wednesday says there are 24 linkage groups, or 24 chromosomes. So there's a discrepancy here. Cavefish can't have both types of numbers of chromosomes. They either have 24 or 25 or fewer. And so part of the paper that's going to be really confusing about on Wednesday, I apologize in advance, is that these authors of this new paper, the Wednesday paper, these are basically ideas of what genes, so each of these is a linkage group, each vertical set of lines, it's linkage group 1, 2, 3, 4, 5, 6, and so forth. The authors have their prediction based on the genome sequence of what genes make up which of these linkage groups or chromosomes. But this paper says 24, the previous publication said 25. And so the prior authors' ideas of which genes are on which chromosomes do not entirely agree with this paper's. So some of the terminology in here, which you don't really have to focus on too much, they're going to be talking about, well, linkage group 24 or linkage group 2 in reference X, we think is the same as linkage group 3 and linkage group 17 in reference Y. Don't worry about that. They're just arguing over what genes go on which chromosomes. That's not really relevant. The relevant part is what genes cause eye loss. Okay, already mentioned pleiotropy briefly. There are two different ways to get this effect. So again, the idea of pleiotropy is you have one gene or one mutation or one change that causes two or more seemingly different changes in a phenotype, like eye loss and pigment loss. I say seemingly different because what if, on the bottom here, there's one gene, say it's a transcription factor that's important in developmental biology that controls both the pathway that leads to eye development and the pathway that leads to pigment production. If you got rid of that gene by mutation or deletion, then you would affect both of those traits, eye development and pigmentation, which maybe at first blush don't seem like they're connected, but they actually biologically are, because at some point every biological process is connected to genetics. So that's one way to get this loss or gain of two traits at the same time, eye and pigment, for example. The other other possibility, the other leading possibility would be that there would be two different genes for those two different traits, but they'd be in the same part of the same chromosome. So that if there was a deletion of that part of the chromosome, that would be one mutation. A deletion is one change, but it would get rid of both of those traits at the same time. So there's two different ways to get to the same effect, reduced eyes and loss of pigmentation. So we're going to see on Wednesday which of those the authors favor. And we don't need to talk too much about this transposable element bit because there are a lot of transposable elements in the genome. As you can see here, they're all lit up in the human genome by yellow fluorescence. So transposons litter our genome. These are pieces of DNA that copy themselves and then paste themselves back into our chromosomes. So there are literally 1 million copies of this 300 base pair long DNA sequence called an ALU sequence. So let's see, 1 million base pairs out of 300, so it's like a tiny fraction of our genome, but still it's a sizable bit of our chromosomes of this junk DNA. This is literally junk DNA. So we don't need to go through this. This is just a cartoon of how retrotransposition works. You get expressed, reverse transcribed, cDNA, and then the cDNA pastes itself back into our chromosomes. But then again, transposable elements is not a huge part of this paper. So if you blinked and missed it, that was okay. And last, we'll do blasts some other time. It's important to learn, but like I said, they did not spend much effort. It was a very small part of the paper that touched on blasts. So we'll talk about this some other point. Yep. So any questions, concerns, comments, gripes, criticisms? Come back Wednesday. We'll talk eye loss. Wear safety glasses.